Basics of Biomedical Instrumentation course. Today, we are going to discuss electroencephalography. It means EEG. Definition of EEG. EEG deals with the study of electrical activity of the brain. The brain waves can be picked up and recorded by the electrodes attached to the skull of a patient. Voltages on the surface of the brain are about 10 millivolts, which propagating through skull bone voltages are estimated to the levels from 1 to 100 microvolt. The EEG electrode pick these voltages. They are in the frequency ranges from zero point 0.5 to 3000 Hz. During recording, electrodes are placed around frontal lobe, parietal lobe, temporal lobe, occipital lobe of the brain. Action potential of the brain. Progressive transient disturbance of the resting potential along a nerve to transmit information from one end to other. The action potential is caused by a very rapid change of membrane permeability to sodium ions. Here we have EPSP and IPSP. The EPSP is excitatory postsynaptic potential and IPSP is inhibitory postsynaptic potential. And third one is effect potential, which means event related potential. Actually, every activity, the membrane will generate a electrical activity, the nerves will create that type of potential, the type of action potential can be further classified as EPSP, IPSP, and evoked potential. Let us see what is EPSP. If the transmitter substance is excitatory, the receptor member potential increase in positive direction. Let us see the graph. See here the EPSB, which is nothing but the receptor membrane potential increases in positive direction, so that the receptor neuron is more likely to discharge and produce a spike potential, and this potential change is called excitatory post synaptic potential. These move the cell towards the threshold by allowing the positive ions to enter the cell. The larger EPSP, the more likely action potential is to fire. Next, IPSP. The IPSP, if the transmitter substance is inhibitory, which means the membrane potential of the neutron receptor, which is neuron, increases in negative direction. See here, in EPSP, it is in positive direction. In IPSP, the potential which is increases in the negative direction, so that it is less likely to discharge, and this potential change is called inhibitory postsynaptic potential. This moves the cell away from the threshold due to the movement of negative ions into the cell. Third one, evoked potential. It is also known as a event related potential. Evoked potential are the potentials developed in the brain as the response to external stimuli like light, sound, etc. By the observance, if you see a light, if you hear the sound, the potentials will be generated in the nerve. That type of potential is based on a stimuli, that is the external stimuli. This is called as evoked potential. The external stimuli are detected by the sense of organs, which cause changes in the electrical activity of the brain. This is known as evoked potential. Right. We have a classification of brain waves: alpha waves, beta waves, theta waves, and delta waves. I told a set of frequency ranges which is available to pick this electrical activity. So, first we go with alpha waves. Alpha waves, which is nothing but the frequency ranges from 8 to 13 Hz. What is the occurrence? It should be found in a normal person if they are awake in a quiet resting state. It may be occur at the occipital region. During sleep, they disappear. So the alpha waves have an amplitude of 20 to 200 microvolt, which means a 50 microvolt. This is the alpha wave diagram. So the frequency range is 8 to 13 hertz. Next, go with the beta waves. The frequency occurrence should happen from 13 to 30 hertz. The occurrence, these are the recorded from the parental and frontal regions. Previously, we get it from the occipital. Here, parietal and frontal regions. This will be recorded from the scalp. So here, two types of it further classified into beta 1 and beta 2. Beta 1 is inhibited by the cerebral activity, and beta 2 is excited by the mental activity like tension. So these are the beta waves. Next, go with theta waves. It may occurs in the range of 4 to 8 hertz. Actually, these recorded from the parietal and temporal regions of the scalp of the children. These also occur during emotional stress in some adults, particularly during disappointment and frustrations. So this can be recorded in the theta waves, which is from the parental and temporal regions of the people or subject. These are the waveforms is nothing but theta. Finally, go with the delta. The frequency ranges from a zero point 0.5 to 4 Hz. The occurrence only once in every 2 or 3 seconds because these occur in a deep sleep in premature habits and in very serious organic brain diseases. So the delta may be occurred in the ranges of zero point 0.5 to 4 Hz. Let us go with the recording setup of EEG, which means an international standard. They devised a 10 to 20 electrode scheme for recording electrical activity of the brain. Now we're going to see, please change in PPT it may wrong as ECG, but we are dealing with EEG, electroencephalography. In electroencephalography EEG, electrodes are placed in a standard positions on the skull in an arrangement called 10 to 20 system, a placement scheme which is devised by International Federation of Societies of EEG. Let us see the diagram. Assume this is the skull of the human. So now I'm going to tell the steps, the procedures to record the EEG signals. As I said, there is a International Federation Society devised a scheme which is called 2020 electrode system. In the examination, if they are asked draw a montage system or 10 to 20 electrode system, then you have to explain these points. Let us go with the first point. First, draw a line from the nation to Union. This area is said to be a nation. This area is said to be a Union. So, just draw a line on the skull from nation to Union. Draw a similar line from a left preacular, that is ear point, to the right preacular ear point, which is nothing but A1 and A2, which is nothing but nation and Nia. The center we may have the intersection of the two lines, which is nothing but CZ. So actually these arrangements it's commonly devised. So for the examination purpose, I'm telling that draw a circle, nation to India, and left preacular point to the right preacular point. So mark the intersection of these two lines, which is called CZ, is the midpoint of the distance between the nation and India, and distance between the auricular points. Now we have to mark the points, right? 10, 20. Why this system is called 10 to 20 recorded system? Because here we have to measure a 10% or 20% distance, we will place the electrodes. That's why it is called as 10 to 20 system. Let us see how. Mark the points at 10, which is nothing but 10 percentage of distance. That is from the neon, which may be nothing but Nashon, which is 10 percentage FPZ. Remaining FPZ to FEZ or FEZ to CEZ or CEZ to PZ or PZ to OEZ, which is nothing but 20 percentages. That is 10, 20, 20, 20, 20, and 10. So the total nation to Indian distance, which is nothing but it's a point of FPZ, FEZ, CEZ, PZ, and OEZ. Next, mark the points from A1 to A2, which means the distance between the preocular points, which means A1, we have 10 percentage distance. First point is nothing but T3, this temporal point. Next C3, which is 20 percentage. Another CZ, which is 20 percentage. Another C4, it is also 20. T4 20, T4 to A2 10. So the total distance between preocular points is 10, 20, 20, 20, and 10 percentage. These points are T3, C3, C2, C4, and T4. In this, we have odd number points. Note these have. On the left hand side, we may mention the odd numbers. That is nothing but 1, 3, 5. In right hand side, we may mention even number of points, which is 2, 4, like that. Here, odd points, which is nothing but T3, C3, and even point, which is nothing but C4, T4, which is on the right. Next, from that, first we finished. Next, we followed with the measure the distance between FPZ and OEZ. So you have a great circle which is passing through the T3 and mark the points from 10, 20, 20, 20 to 10 percentage. So OZ to O1 you have 10 percentage, FPZ to FP1 we have 10 percentage. Rest of them we have 20 percentage. So these are the positions from the odd side. So we will be numbering at FP1, F7 and T5 and O1. Same procedure will be followed on the right hand side and mark the position which is nothing but 10% from FZ to FP2 and OZ to O2. Rest of the positions have the 20 percentage. So we mark FP2, F8, T4, T6 and O2. Now this is the diagram for the 10 to 20%. The next point we have to measure the distance between FP1 to O1. So along the greater circle, we have passing through the circle C3, we may have a points of position F3 and P3. The ground reference electrode is a metal clip at the earlobe. So repeat these procedures on the right hand side, then we may measure F4, C4 and P4. Now we may check the F7, F3, FZ, F4 and F8. These are the equidistance along the transverse circle passing through F7, FZ and F8. And check the point T5, P3, PZ, P4 and T6. These are the equidistance along the transverse circle which may pass through T5, PZ and T6. So before placing the electrode, the slag should be clean and lightly abraded and electrode paste is applied between the electrode and the skin, which means the application of the electrode paste, the contact impedance which is less than a 10 kilo ohms. Next, we will see the modern EEG unit. Let us see the diagram. This diagram shows the modern 8-channel EEG recorder. The patient cable, which consists of 21 electrodes, are connected to a 8-channel selector. We have 21 electrodes. These 21 are connected to the 8-channel selector. These electrodes are attached to the channel selector group of 8, which is called montage of electrodes. So the left ear electrode acts as the reference electrode of the left brain electrodes. The EEG unit is placed in the room which covered the ferrous metal screen to reduce the 50 Hz of AC alternate current interference. This interference is reduced by employing differential amplifiers as a preamplifiers. So the filter bank consists of appropriate filters to select these different types of brain waves: visual stimulus, audio stimulus, tactile, that is which means touch stimulus, which are used to record evoked potentials from sensory part of the brain. So the time delay between the stimulus response and can be measured in the signal processing unit. Next, the input should be passed to the amplifier. The amplifier is further divided into two, that is pre-amplifier and driver amplifier. Both amplifiers are split into different stages: stage 1, stage 2, stage 3, stage 4, stage 5. These stages are cascaded to increase the gain of the pre-amplifier and driver amplifier. Now, the amplifier input is passed to the writer, behaves as the storage memory. So, further, it should be passed through the traces which process the signal and which can be displayed through the output unit. Now we have to compare the input, amplifier, writer, and tracer. Input, what is the input? Visual, audio, task stimulus. Amplifier, differential amplifier bank. Writer, which is nothing but a storage memory. Trace, which is nothing but signal processing data, and CRT display. Now we go with analysis of EEG. Right. Electrode scheme we completed and we devise the block diagram of EEG. Now, after picking the EEG signal, what are the analysis will go on? What are the applications will be going on? Let us see the level of consequences. EEG changes with the level of consequence. The lower frequency and larger amplitude has diminished mental activity. So the REM sleep here mentioned as awake, light sleep, REM sleep, deep sleep, and cerebral death. The REM sleep coincides with the periods of dreaming. As anesthesia is applied in the brain, frequency decreases, amplitude increases. Thus, theta and delta waves appear. So, in case of cerebral death, that is which is called brain death, EEG shows the permanent absence of brain wave even through respiration and circulations are maintained, which is nothing but a straight line. There is a no activity which is recorded in the brain. That is nothing but brain death. Next, brain tumor. The tumor displaces the cortex, and if it is large enough, of the electrical activity will be absent in that of hemisphere. If a tumor occur in the particular part, the part of the hemisphere there is no electrical activity will be observed. Since no electrical potentials originate in the tumor itself, which is damped EEG, is the sign of tumor can be observed. Next, epilepsy. It's a symptom of brain damage. This may due to the defects in birth, delivery, or head injury, or during accident or boring. So it may also due to the brain tumor, the seizures may happen. This is the epileptic seizure EEG wave. Thanks for watching the session. So far, we well discussed what is EEG, how action potentials of EEG may be recorded, EPSP, IPSP evoked potentials. Next, how recorded the EEG signal by means of 10 to 20 electrode scheme, which is nothing but montage electrode scheme, and block diagrams, simplified block diagram of EEG, and how EEG signal should be analyzed. Thanks for watching the video. Thank you. In the next class, we will discuss electrocardiography, which is ECG.